This video will be on mitosis and meiosis. So first we're going to start out talking about the cell cycle. The cell cycle is the time from when a cell is first born from a previous cell division to when it completes another division. And the cell cycle is divided into two parts. There's interphase and there's M phase. M phase stands for mitosis, and interphase includes three primary steps. When a cell is first born, it will enter into G1 phase. G stands for gap phase, and during G1 phase, the cell undergoes significant growth. Sometimes a cell will decide to withdraw from future cell divisions, and if this happens, it will actually enter into this alternative pathway called G0. After G1, it enters into S phase. S phase is when the DNA is replicated, and we will talk about the process of DNA replication in quite a bit more detail later in the course. And then after S phase comes G2. G2 is primarily preparation for mitosis. At the end of G2, the cell will enter into mitosis, and mitosis consists of two main important things. The first thing that has to happen is the chromosomes have to separate, and following the chromosome separation, the cell itself has to separate, and that is called cytokinesis. Now in this course, our main focus is actually going to be not on mitosis, but on meiosis. We're going to talk about the stages of meiosis in a minute, and we'll talk about the fact that meiosis consists of two cellular divisions going from 1 into 4 cells. I'd just like to make the point now that meiosis is also preceded by this same type of cell cycle. Even though there are two cellular divisions in meiosis, it is only preceded by one cell cycle. And so a cell that is going to enter into meiosis would go through G1, go through one S phase, then G2, and then it would do one division of meiosis followed by the second division of meiosis. Next, we're going to talk about chromosome structure a bit and what happens during S phase. So eukaryotic chromosomes are linear, and they have centromeres. Centromeres are the place where the kinetochore and the astromicrotubules will attach to separate the chromosomes during cellular division. And the centromere doesn't have to be at the middle, however. It can be near one of the ends or in the middle. At the ends of the chromosomes are the telomeres. So during S phase, or DNA replication, that one DNA molecule, this is a double helix DNA molecule, is replicated to make two double helix DNA molecules, and those are shown here. Those two are exact copies of each other, and they are called sister chromatids, or often we just call them sisters for short. Now at the end of S phase, those two sisters are still attached to each other, and they are attached at the centromere. So even though these are two complete DNA molecules, we still only call them one chromosome as long as those two sisters are still attached to each other. Let's talk about the stages of mitosis next. So remember that as a cell is getting ready to enter into mitosis, S phase has already happened. And so in the nucleus, what you have is actually sisters that are attached to each other. You can't see the structure of these since the DNA is not yet condensed. Mitosis consists of 4 steps: prophase, metaphase, anaphase, and telophase. Now some sources, including your textbook, separate out another phase called prometaphase. I'm not actually interested in having you learn the details of prometaphase, so everything that happens in prometaphase in your textbook, we're going to call late prophase or just prophase. So if we look at what starts to happen in prophase, the nuclear envelope is going to start to break down and the DNA condenses. And near the end of prophase, each of those pairs of sister chromatids are going to be captured by the spindle fibers that are emanating from the centrosomes, or the poles of the cell. Next comes metaphase, and during metaphase, those sisters line up along the equator of the cell, which is also called the metaphase plate. At this point, each of those pairs of sister chromatids have a spindle fiber going in either direction. So each pair has been attached to both of the spindle poles. In anaphase, the connection between those two sister chromatids breaks down, and the sisters release from each other and begin to get pulled toward separate poles of the cell. Finally, in telophase, the chromosomes reach the poles and the nuclear envelope begins to reform. The DNA will then decondense, and at the very end of mitosis, cytokinesis will occur. So, to summarize the key points of mitosis, first of all, mitosis is asexual reproduction. It is just how cells divide to form two cells from one cell. This occurs in somatic cells. Somatic cells are any kind of cell that is not a germ cell. So a germ cell, those are the ones that produce the gametes, the egg and the sperm. And finally, mitosis produces two cells that are genetically identical both to each other and also genetically identical to the mother cell that produced them. Now let's talk about meiosis. So meiosis involves sexual reproduction, and sexual reproduction really has two main aspects to it. First of all, the actual process of meiosis, which is how haploid cells called gametes are formed, and then fertilization. So two gametes will join together to form a fertilized embryo, creating the next generation. The consequence of sexual reproduction is a large amount of genetic variation. So I'd like to start off with an overview of meiosis. Meiosis consists of two divisions, Meiosis I and Meiosis II. They are preceded by an interphase with G1, S, and G2, as I previously mentioned. The first division of meiosis involves something very unusual. What happens here is that homologous chromosomes locate each other in the cell. And I want to point out that in mitosis, homologous chromosomes completely ignore each other. But meiosis, here those homologous chromosomes find each other and they line up. And then each of those pairs of sisters that are the homologous chromosomes separate from each other into the two separate cells. Meiosis I is also called a reduction division, and the reason is that if you actually count the number of chromosomes at the beginning compared to the end of meiosis I, you'll see that the number of chromosomes per cell actually decreases in number. Meiosis II is very, very similar to mitosis. So here, those pairs of sisters will just line up on the equator of the cell, and the sisters will separate from each other. Meiosis II is also called the equational division, and in this particular division, you do not decrease the number of chromosomes per cell. In this particular figure, you started out with 2 chromosomes per cell, and you ended up with 2 chromosomes per cell. Meiosis I is broken down into the 4 stages, prophase, metaphase, anaphase, telophase, as is meiosis II. When we talk about prophase in meiosis I, we're simply going to refer to that as prophase I, whereas if we were talking about prophase in meiosis II, we'd call it prophase II. So prophase I is a very, very long step and a very complicated step. Scientists have actually broken this down into a number of discrete steps called leptotene, zygotene, etc. You don't need to learn these particular names, but I do want you to know all of the events that occur in prophase I. So some of the stuff we've talked about before also happens here. So the nuclear envelope breaks down, the chromosomes condense, and this is the point where the homologous chromosomes are going to line up with each other. Again, remember that S phase has already occurred, so those chromosomes are present as pairs of sisters. Those homologous chromosomes will progressively line up along their length, and the process of these homologous chromosomes lining up is called synapsis. Once synapsis is complete, you have something called a tetrad. Tetrad is the preferred term that I like to use. Other people also call it a bivalent. Those two terms mean the same thing. This is really a descriptive term because you've got actually four complete double-stranded DNA molecules that have lined up together. You have two sisters from the one homologous chromosome lining up with the two sisters from the other homologous chromosome to make four DNA molecules. The other thing that happens during this stage of prophase I is that crossing over will happen. And so when you have the homologous chromosomes lined up, sometimes they can actually switch their ends. And this switching occurs between the non-sisters, between the homologous chromosomes. And sometimes you actually see these little X's, which is a hallmark of crossing over. And these little X's are called chiasmata. All right, let's go into the stages of meiosis I in more detail. So these top ones are all prophase I. Again, the nuclear envelope breaks down, the chromosomes condense, the homologous chromosomes line up, and you form these tetrads. These tetrads can undergo crossing over, and toward the end of prophase, they will start to be captured by the spindle fibers that are emanating from the spindle poles of the cell. In metaphase I, the homologous chromosomes, the tetrads, will line up along the equator of the cell, and each of those tetrads is captured by two spindle fibers, one from each pole. In anaphase I, the homologous chromosomes will separate from each other. And I want to point out that the sister chromatids do not separate from each other in anaphase I. In telophase, those pairs of sisters will reach the poles of the cell. The chromosomes will very briefly reform that nuclear envelope and decondense, and cytokinesis occurs, forming two cells and completing the first division of meiosis. Those two cells will enter a very brief period of interphase. That period of interphase is not a normal cell cycle. There is not another S phase before they enter right into meiosis II. Meiosis II, very similar to mitosis. In prophase II, the nuclear envelope breaks down again, the chromosomes condense, the sister chromatids begin to get captured by the spindle fibers, and in metaphase II, those sister chromatids will line up on the equator of the cell. In anaphase II, the sisters will now separate from each other, just like what happens in mitosis. And in telophase II, those individual chromosomes reach the poles of the cell, nuclear envelope starts to form again, and cytokinesis occurs. The DNA will decondense. You end up with four cells, and each of those cells are a haploid number of chromosomes. So just to compare and contrast what happens during mitosis to what happens during meiosis, meiosis is sexual reproduction. At the end of meiosis, you end up with gametes. This would be the egg and the sperm in animals. These cells have a haploid number of chromosomes, or a 1N number of chromosomes, and very importantly, every single one of those is genetically different from each other, and they are also different from the mother cell that produced them.